Okay, great. Thanks everybody for coming. I'm excited to share some of the work that I've been doing as part of my postdoc in the Columbia lab, working on host symbiote interactions at the genetic level. So just broadly, for species that that harbor symbionts, either as a specialized interaction or more as a microbiome, we see these patterns where essentially the symbiont and the host genomes need to mesh, or they need to be able to interact in order to exchange nutrients or provide defenses or whatever the actual mutualism is based off of. And we can see that even at the sort of broadest levels. So in these wasps, we can see that phylogenetic relationships of the insect hosts predict the relationships of the microbiomes that they're hosting. And so even when they're hosting these large consortia of microbes, the sort of genetic underpinnings of the interactions between the host and the symbionts can be driving the structure of these symbiont communities. And in my system, in coral reefs, we think about these interactions both in terms of who is going to interact with whom. So corals have to form endosymbiotic interactions with these algal symbionts. But of course, when you arrive at a reef as a newly recruited coral, you're faced with the prospect of potentially interacting with lots and lots of different symbionts. And we know at the genetic level that this is true, that the symbionts are highly genetically diverse. And it's a diversity that appears to match the diversity of the corals with whom they associate. And so we might think that as you're forming these interactions and as these interactions are persisting through time, there might be more optimal partnerships at the genetic level that really enhance the ability of the holobiont to perform optimally for a given environment. And I'll just point out here that this is a good example of a reef that we work on in the Columbia lab. And as you can tell, the environment can shift just over the course of a small reef that you're looking at here. So there are places of this reef that are going to be a lot different environmentally than others. And that's true both at the reef level, but also across the entire geographic range of these species, which can be quite extensive. And of course, this is important not only for forming the optimal partnerships, but also for forming partnerships that are stable in the face of marine heat waves that are becoming more and more common, especially in these tropical systems. And so what this picture is meant to emphasize is that we have two colonies here that are clearly experiencing similar environments. They live in the same place, they're next door neighbors. And yet when things got warm, one colony bleached out, so the association between the host and the symbiont dissolved, while the other one remained perfectly healthy. And so a large question that we're focusing on in the lab is why do we see this discrepancy? Why, when you experience the same set of stressful conditions, do some partnerships maintain their ability to perform in the face of those stressful conditions and others don't? And so we've been focusing on that at the genetic level as part of a broader study on Acropora hyacinthus across the Palauan archipelago. And this is part of a broader study meant to figure out where heat-resistant colonies reside, whether or not there are geographic patterns associated with that. And I would point you to this paper that we published last year if you want to know sort of the broad scale picture of where we find bleaching resistant corals and where we don't. But for our purposes today, what I'm going to do is focus just on that bleaching resistance in the context of the interaction between the host and the symbiont. So I'm going to ignore the environmental impacts on this system for the moment, but I'd just like to highlight that they are really important and highly variable. And so what I'm going to tell you today is about a story for about half of the colonies we studied as part of this larger study, where we sequenced 200 host and symbiont genomes to start genotyping them across the entire genome to figure out whether or not we can detect these associations or matching patterns between host and symbiont alleles. And so what I mean by matching is looking for combinations of alleles that are beneficial in stressful conditions for the host and the symbionts. So all of these corals have been experimentally heat stressed. So we know that when we take them up to a certain temperature, we do that for every colony as part of the study. We know how many symbionts they're likely to retain and how many they are going to lose. And so in combination with that, we also know their genotype across lots of different loci. And when we look at these, what I just want to do is show you an example of this for a highly correlated or a locus combination that we're calling well matched. And what you can see is that if we know something about the genotype of the host, we know something about the genotype of the symbiont too. So for the most part, both of the host and symbiont genotypes in this example are homozygous for the alternative allele. So they're following this upper right-hand quadrant of this graph. So the symbiont genotype is a 1. So that's a frequency of 1 for that alternate allele. And same for the host genotype. However, when one of the two partners is a heterozygote, we see that more often than not, its partner will also be a heterozygote. So if the coral has a heterozygous genotype for this locus, we see that the symbiont tends to as well. And what I've done here is just draw a best fit line of a linear model predicting the host and the symbiont genotype based on those allele frequencies. However, what we can see is that there are some individuals that are following the global pattern and others that are not. And so what I've done here is just highlight 2 hosts that are more or less matched based on that global pattern that we would predict we should see across all these individuals. So in this instance, this individual is not far from that global line. The residual is another way to put this, is low, whereas individuals up here, these hosts are homozygous for the alternate allele, but they are associating with symbionts that are heterozygotes. And so for this particular locus combination, we would say that those 2 hosts are less well matched than this host based on that overall pattern across all 200 individuals. And the interesting thing that we find is that when we plot those, so those residuals or the degree to which the host and the symbionts are matched, we find that that predicts whether or not they will retain their symbionts when they're experimentally heat stressed. And so what I've done now is shown on the y-axis that deviation, so how far you are from the global pattern of matching or not at a particular locus combination. And on the x-axis, I'm showing the retention score, which is the proportion of symbionts that you are left with based on how many you began with before the experimental study. So essentially, we have a control coral and a heated coral, and the retention score is how much that heated coral matched the control colony in terms of the number of symbionts that are retained at the end of experimental bleaching. And what we can see is that for this locus combination, that deviation or the residual tends to be higher when they are going to bleach more. And when they retain more, that deviation at the genomic level or the match tends to be better. And when we did this across the 31 SNPs that were the most highly correlated in this last round of analyses that we've run, we find that almost universally that pattern holds where there's at least a negative association between the degree to which you're matched and how much you bleach. And that holds for all but 2 of 31 SNP pairs, which is far beyond the 50-50 that we might predict based on a null distribution. And so as you can see from these plots, they're not always the strongest correlations. There's often a lot of noise, but they're almost universally trending in that pattern. And I'll just point out, although I'm not going to talk about it much here, that that is a similar result for the remaining symbiont density. So not scaled to how many you began with, but just what you end with. The overall standing symbiont population after experimental heating also is associated with how much matching you have going on at the genomic level. And we are interested in this because we think that it might help explain why it is that just looking at the coral genotype alone has done little to explain how much bleaching we're likely to see on a reef. So the most extensive study that's done this so far came out a couple of years ago by Fuller et al. 2020. And they basically looked at all these different things that can affect bleaching that you might expect to affect bleaching, like the environment and the proportion of symbiont species that tend to be more bleaching resistant. But what I'd like to highlight here is this polygenic score, is this last bar at the end, explaining the overall variation on the y-axis in bleaching propensity. And this polygenic score, although it did explain more bleaching, it didn't explain much more bleaching. And so we don't actually gain that much more information knowing the genotype of a coral to predict how much it's going to bleach. And that might be because these interactions between the corals and their symbionts at the genomic level are really determining how well they function. And so if they're not well matched, knowing just the coral genotype might do little to tell us about how well they'll perform in the face of stressful conditions. And so just to wrap up, What this is suggesting to us from a conservation perspective is that the variation at the genetic level might be essential for coral survival, and that might be the case both for the host and the symbionts. It might require high levels of genetic variation at both to ensure that these optimal partnerships can become established every generation. And so in this toy example that I'm showing here, this black genotype of coral matches really well with this blue genotype of symbiont, and it works in this environment. And then just to point out that when that environment changes, the background in which those two partners find themselves, there might be a different combination that matches even better, and which highlights the importance of preserving as much diversity as you can, if that's indeed what's mediating the outcome of this interaction. So with that, I'd like to thank the following people, and thank you for your attention.